All right, let's get into it. Today, we're cracking open one of the greatest detective stories in the entire history of science. Seriously, it's a mystery that gets to the very heart of what life is.
I mean, really think about that for a second. What's the secret instruction manual inside you that decides, everything, from your hair color to how your body fights off a cold? For the longest time, that was one of science's biggest unanswered questions.
Well, the answer is this incredible molecule right here, DNA. This is the stuff our genes are made of. But figuring that out? Oh, that's the real story, and that's what we're going to unpack today.
Okay, so let's set the scene. We're talking about the early 1900s. Scientists were pretty sure that traits were passed down by these things they called genes. But here's the catch. Nobody had a clue what a gene was actually made of. Was it a protein? Some kind of complex sugar? Nobody knew, and the race was on to find out.
So our first major clue pops up in 1928, thanks to a British scientist named Frederick Griffith. And the funny thing is, he wasn't even looking for the secret of life. Not at all. He was just trying to figure out how certain bacteria give people pneumonia. But, as it so often happens in science, he stumbled onto something way, way bigger.
So Griffith was working with two different versions, or strains, of the same bacteria. You had the S strain, which was the nasty one. It grew in these smooth little colonies, and yeah, it was deadly. And then you had the R strain. Its colonies looked rough, and it was totally harmless.
Okay, so this is where things get really interesting. He starts his experiments. First, he injects a mouse with the deadly S strain. The mouse dies. No surprise there. Then, he injects the harmless R strain. The mouse is fine. Makes sense. Then he takes the deadly S strain and kills it with heat before injecting it. The mouse lives. Okay, so it's not some poison left behind. But then, he does something kind of weird. He mixes the dead S strain with the live, harmless R strain, injects the combo, and the mouse dies.
Whoa, hang on. How did that happen? The harmless bacteria somehow turned into the deadly kind. And when Griffith checked the mouse's blood, sure enough, he found living, deadly S strain bacteria. So what on earth was going on? Griffith figured that some kind of transforming factor, some chemical substance must have been passed from the dead S strain to the living R strain. This mystery stuff carried the instructions for how to be deadly. And since the newly deadly bacteria could pass this trait on, he realized this transforming factor had to be a gene. He'd found a huge clue, but he still didn't know what the chemical itself was.
So fast forward to 1944. A team of scientists in New York, led by this guy Oswald Avery, decide to pick up this cold case. Their mission was simple: find this mystery molecule, pinpoint exactly what Griffith's transforming factor was. And their method was just brilliant. It was a classic process of elimination. They used special enzymes to destroy just one type of molecule at a time. So first, they get rid of all the proteins. Did the transformation still happen? Yep. Okay, so it's not protein. Then they destroy the lipids. Transformation still happens? Not lipids. They do the same for carbohydrates, and even another molecule called RNA. And every single time, the harmless bacteria still turn deadly. But then came the big one. They used an enzyme that only destroys DNA. They mixed everything together, ran the experiment, and nothing. The R strain bacteria just stayed harmless. The transformation was completely blocked.
That was it. That was the moment. By showing that only destroying DNA could stop the transformation, Avery's team proved it. DNA was the transforming factor. It's the molecule that stores and passes on genetic information from one generation of cells to the next. They had found the gene.
You'd think that would be the end of the story, right? But this was a huge, huge claim. And science is all about skepticism. A lot of scientists just weren't convinced. They still thought it had to be protein. So the world needed one final knockout piece of evidence. And that came in 1952 from two American scientists, Alfred Hershey and Martha Chase.
So Hershey and Chase had a secret weapon. It was this tiny virus called a bacteriophage, which literally means bacteria eater. And the coolest thing about these little guys is how they work. They're like a tiny syringe. They latch onto a bacterium and inject their genetic material inside, leaving the rest of their body outside.
And this is what made them the perfect tool. See, a bacteriophage is super simple. It's just a protein coat on the outside and a core of DNA on the inside. That's it. So it boils the whole mystery down to one simple question. When the virus infects the cell, what actually goes in? Is it the protein or is it the DNA?
And this is where Hershey and Chase got really clever. They used radioactive atoms as little tracking devices. They knew that proteins have sulfur in them, but no phosphorus, and DNA has phosphorus, but no sulfur. So they made two separate batches of viruses. In one batch, they tagged the protein coats with radioactive sulfur. In the other, they tagged the DNA core with radioactive phosphorus.
Then, they let both batches of viruses do their thing and infect a bunch of bacteria. All they had to do was check where the radioactivity ended up. And the result? It was so clear. For the viruses with the tagged protein coats, all the radioactivity stayed on the outside of the bacteria. But for the viruses with the tagged DNA, almost all the radioactivity was found on the inside.
That was the smoking gun. It proved, beyond a shadow of a doubt, that the virus injects its DNA, not its protein. Their experiment was the final nail in the coffin. It convinced everyone that DNA was the genetic material for all living things on Earth.
Okay, so case closed. DNA is the stuff of life. But finding that out just opened up a whole new set of questions. Like, what does DNA actually have to do to be the molecule of life? Well, scientists realized it had to have three absolutely critical jobs. First off, its main job is to store information, a lot of information. DNA is the master blueprint. All the instructions that make a person or a plant a plant, it's all written in the chemical code of DNA.
Second, you've got to be able to copy that information, right? Every time one of your cells divides, it needs to make a perfect, complete copy of its entire DNA blueprint to give to the new cell.
And third, that information has to be transmitted or passed on. Genes are handed down from one generation to the next. So that means DNA has to be packaged up and passed along very carefully when organisms reproduce.
You know, a really great way to think about it is like a treasured family book. The information inside is priceless, so it has to be stored carefully. It needs a way to be copied so the information can be shared. And of course, it has to be tough enough to be passed down through the generations. That's DNA. Figuring out that DNA is the molecule of life was easily one of the most important discoveries of the last century. It opened the door to everything we know about modern biology. And it leaves us with this huge, amazing question to think about. Now that we've actually cracked the code of life itself, what are the limits to what we can do with it?